Today's speaker is Dr. Mark Levern. Mark is the Vice President of Certara Strategic Consulting, where he leads and contributes to numerous consultancy projects involving population PK, PK/PD, and model-based meta-analysis. Mark earned a PhD in biomathematics from the North Carolina State University, and his most recent therapeutic area experience has been in therapies for infectious disease, metabolic, and autoimmune disorders. Mark, welcome to today's presentation. I'll now turn it over to him to begin the webinar.
Appreciate the chance to speak with all of you. Just so you know, this is I'm giving today an encore presentation of a talk I gave at last year's ACOP meeting. So if you happen to attend that session, this may look a little familiar to you. But I think it's relevant beyond just the audience that we saw that attended that day, and so we thought it was worthwhile presenting for a Certara webinar. So at a glance, this is my agenda for the day. We're going to talk about, first of all, what is model-based meta-analysis, how it can, at a conceptual level, provide valuable information for decision making, especially in this case focusing on autoimmune diseases. So the initial part of the talk is really more of a conceptual introduction, and then the remainder of the talk will just be talking about some case studies in which the approach is applied to drug development in autoimmune for autoimmune therapies, including how it's been used to compare drugs in therapeutic classes, optimize clinical trial designs, predict long-term responses based on short-term endpoints or biomarkers, and then also how it can potentially be used to guide dosing recommendations for unstudied indications or populations. So, what can model-based meta-analysis do to help drug development in autoimmune diseases? Well, essentially one thing it can do is it can help abbreviate the costs of drug development. And by that I mean, if you look on the right-hand side, essentially I like to call this the spiral of cash, in the sense that this is money and time and resources devoted to developing your own proprietary data. And generally it's a cyclical process throughout drug development, in that generally you'll start by designing an experiment or trial. You'll conduct that trial, you'll analyze those results, and then you'll use those results to potentially make inference about your drug, and then possibly design another trial or eventually apply for the approval of the drug. And every iteration through that cycle requires a lot of spending on the part of the sponsor. So on the left is the idea of essentially augmenting this cycle of generating proprietary data by leveraging public and preclinical data that's already out there available for your use. So as an example of this, here is a plot, and the title pretty much says it all. If you were to generate the data that was shown in this plot, it would essentially cost your company about $7 billion. So what are we looking at? Now this is not, as a caveat, this is not an autoimmune plot. This is really more in the metabolic area. And on the x-axis we have change from baseline in fasting plasma glucose, which is a very rapidly responding biomarker. And on the y-axis, we have change from baseline in HbA1c. The colors in this plot all represent different therapeutic classes for against essentially anti-diabetic therapies. And what this plot is showing is that HbA1c, the longer responding endpoint, is highly correlated with a very rapidly responding endpoint, and thereby can be largely predicted based on a short-term response. Now, why is this a $7 billion plot? Well, essentially, there's 57 drugs represented in this plot over 9 therapeutic classes. And if, again, you were to generate all of this data by yourself, assuming that the development of one anti-diabetic therapy is about $200 million, essentially the math works out that if you had to generate this all on your own, it'd be about $7 billion. Now, of course, no one would ever do that in the real world, but this is just a conceptual example illustrating how leveraging of publicly available information can be a big cost savings and a big perk to informing your drug development decision making. So, how does model-based meta-analysis compare with other forms of analysis? People have been doing meta-analysis in the pharmaceutical industry for decades, and so it's probably helpful to understand how this all fits together. So probably the earliest examples of meta-analysis are the simple pairwise meta-analysis. And essentially what this is if you had multiple trials in which drug A was compared to drug B, in essence what you do in pairwise is you essentially put those individual trials together as if they're one big trial. Of course, there are some subtleties in that you have to appropriately weight those observations both based on the sample size and also their variability, and then maybe you consider a few other factors such as like baseline characteristics of your population. But the key concept is that you're only comparing things that were at some point compared together in the same trial, and therefore you're combining that information across trials, but in every instance drug A was compared to drug B in all of these trials. So an extension of pairwise meta-analysis was network meta-analysis, which essentially, using some assumptions about consistency across trials, allows you to bridge and make comparisons between two treatments that may never have been studied together in the same trial. So for instance, let's assume we want to compare drug A to drug C, but drug A and drug C were never studied together in an actual trial. But there are trials in the literature in which drug A is compared to drug B and drug C is compared to drug B. And in that instance, you can essentially use B as a bridge comparing A and C. So literally it is a network in that you're using these connections to compare treatments that may never have actually been studied together in the same trial. So in essence, model-based meta-analysis is an extension of network meta-analysis insofar that we are using a network information structure, and that is meaning that we can compare treatments that are not found together in the same study in the real world, but then we add to that the ability to essentially incorporate pharmacological information, so dose-response relationships and time dependencies and other concepts that we incorporate into pharmacology is incorporated into the analysis. So that's, in essence, how model-based meta-analysis represents an extension of network meta-analysis. So now, as I mentioned, let's go through a few examples of how this approach has been applied in autoimmune diseases. So first of all, the first model-based meta-analysis that was published in Rheumatoid Arthritis was in 2011. It was published by Mandama et al. Yacht Mandama is, of course, with our company, but this is the first published model-based meta-analysis. Prior to this, there were other meta-analyses published in the literature, but one of the things that they did not incorporate were elements of dose response. So here you can see in this plot that this model was based on a dose-response approach. And also what the authors did is they simultaneously fit ACR 20, 50, and ACR 70 responses as an ordered categorical variable. So each of these endpoints is essentially a different level of response, which ACR 70 means you've essentially had a 70% reduction from baseline in your ACR score. And so therefore the most stringent and therefore the least likely. And so they successfully described this dose-response relationships across several therapeutic classes, largely with regard to monoclonal antibodies, although others were also included. And then used this analysis to do essentially competitive comparisons across the various drugs. So the next instance we have in the literature of a model-based meta-analysis in autoimmune was again in the RA indication, and was published by Hamrit et al. In 2012. And essentially, they followed somewhat what Mandema et al. Had done in their initial work, but they then extended it further in that they incorporated time dependencies into their analysis, whereas Mandema et al. Simply looked at time points 12 weeks and later with the assumption that at those later times the disease and drug response were fairly stable. But here, the authors actually incorporated time dependence, and then essentially what they did is they created models that would explain etanercept and adalimumab time course with regard to ACR 20 response. And then what you see in the beige shaded with the yellow squares with the arrow bars, this is the proprietary data that actually came from their trial. And so in essence, what they were able to conclude from this analysis is they were able to position their drug relative to both etanercept and adalimumab as an RA therapy. And for both comparators, they concluded that it was unlikely that their drug would perform as well or better than either of their comparators. And based on this, the weight of this analysis, it was decided to terminate the program, which of course allowed the sponsor to reallocate resources to an asset which was more likely to be successful. So in 2014, our next example was published, and this was essentially applying a model, essentially a model-based meta-analysis approach to clinical trial simulation. And essentially, again, a model was developed based on a literature database, and then essentially the model was used to simulate the planned dose-ranging design that the sponsor had in mind. And to be clear, you can see on the lower part of this plot, you can see that the treatments that were originally planned were 25 milligram, a 100 milligram, and a 200 milligram given at on day 0, week 4, and at 16 weeks. In addition, they had planned to run a 200 milligram every 4-week treatment arm. So what the trial simulations indicated was that if they were really trying to do dose ranging, that 200 milligram Q4 weeks was not very informative, because in general, almost every simulation that was done, it was essentially that regimen would be at the top of the Emax curve. So eventually it was decided to drop, based on this analysis, to drop that last regimen and instead add a 5 milligram given at the same 0, 4, and 16-week schedule, which, as you can see, the yellow indicates that regimen is probably somewhere between placebo and plateau, and the red means that it's clearly in a subtherapeutic area, which would give a much better estimate of dose response. So this design was utilized instead of the initial design, which would have incorporated something that was a regimen that wasn't terribly informative. So then in 2016, we have another publication by Wang et al. And in this publication, they demonstrated that ACR 50 response at 6 months was highly predictable based on ACR 50 response at month 3. And so clearly, this was, again, a model-based analysis, but I think it's fairly apparent what the value proposition is here. If you can shave 3 months off the duration of your trial and have a pretty good idea of what's going to happen at 6 months based on what happened at 3 months, it's that much more quickly that you can either move ahead with your drug or potentially, realize that maybe the drug isn't going to perform at the level hoped, and therefore let's move on to the next candidate. So I think that particular plot largely speaks for itself. So then my final example of an application today is something that this actually is not published data, but extensively anonymized for conceptual, a conceptual example. But imagine, if you will, that a drug has been approved in psoriasis, and now the sponsor wants to move into other indications such as psoriatic arthritis and ankylosing spondylitis. Well, in the case of psoriatic arthritis and psoriasis, they often have a shared endpoint that's reported in both types of trials. Specifically, they often will both report the PASI score, even though in psoriatic arthritis, that's probably not the key endpoint of primary interest. So what we can do with that is essentially we can bridge across these various endpoints and essentially develop a dose response model that describes the key endpoints across all 3 indications, and we can formally test whether or not there are differences in potency across those indications. And then essentially what the vertical lines represent are planned regimens that the sponsor intended to look at for both the psoriatic arthritis and ankylosing spondylitis indications. And you can see what this analysis gave some assurance about is that in general, across these indications, the dose regimens are appropriately spaced to give some feeling for what dose response looks like in these indications. So, as I said, this is a particular example is pretty heavily anonymized, so I will not go into further detail. But I hope I've given you, at a high level, some feeling for how model-based meta-analysis, well, first of all, what it is, and what value potentially it can bring, and how it differs from more historical forms of model-based meta-analysis. And then in the second portion of my talk, hopefully I've given some ideas as to, in general, how the approach has been applied to specific applications in autoimmune drug development. That's, thank you for your attention. This concludes my talk. I think now we can take questions from the attendees.
Okay, thank you so much, Mark. As Mark mentioned, he is ready for your questions, so please do enter your questions for Mark into the Q&A box. Looks like we've got our first question. Mark, somebody would like to know, can model-based meta-analysis be applied to other therapeutic areas besides autoimmune disease?
Oh, yes, absolutely. We've, even just with, we're just but one contributor to this field, but within our company, we've applied it to neuropathic pain, to metabolic diseases such as diabetes, cardiovascular, and also chronic kidney disease. So there's a wide variety of applications. It really, the key limitation is how rich the literature is on a therapeutic area. Of course, the more sort of cutting edge the therapeutic area, the lot more likely that the literature is going to be emerging and therefore not as rich. So there may be some limitations about what you can do, but particularly in the established therapeutic areas, there's just a gold mine of information that could be, leveraged, I guess is the word I would use.
Can you discuss the process you use to prepare data for doing model-based meta-analysis?
Yeah, so it all really starts with data, and essentially a major part of the effort in any of these types of analyses is extracting all of the literature information or information for the clinical trial registries. That has to be turned into a tabular, analyzable form, and in addition, there has to be a lot of what we call augmentation, which is to some degree harmonizing how things are reported with regard to endpoints. For instance, in some therapeutic areas, you may find that two endpoints might have a very similar naming convention, but have very different interpretations or definitions. So there's a lot of work between simply extracting the literature sources and then formatting the data and preparing it for an analysis. And then from there we go into a exploration of the data, and then finally to actually analyzing it with a parametric model. So there's in terms of effort, a good 70% of it is in extracting the data and then essentially getting it in an analysis ready format, which is one of the reasons that as a company we offer databases across a variety of therapeutic areas that are specifically developed in mind for those that want to apply them to doing model-based meta-analysis.
Okay, we have another question. What is your recommendation for key weighting factors to be included in the analysis?
So yeah, as far as weighting, you definitely want to take into account the sample size and the variability. In some instances, there's a measure of variability is often not reported in the literature, in which case our typical practice is to develop an imputation model that whereby if the author doesn't report like a standard deviation, that usually we can get a pretty good estimate of what that standard deviation should have been based on, for instance, baseline, time, and other potential explanatory variables. But we do try to take both of those into account, both the sample size and the variability in weighting the model fits. As far as other factors to consider in your model, such as covariates, baseline status is one that's almost always showing up in our models because oftentimes the worse your baseline status is, the more room you have for improvement, and therefore the more opportunity for it to see a treatment effect. The only caveat is that oftentimes since you're working with summary mean data, you may often not see a lot of dependence on factors that you would see if you're looking at individual data such as body weight or age, because oftentimes those things are going to be fairly consistent across trials in the same population.
Someone wants to know, can MBMA be applied on clinical trials that use different but related or correlated endpoints?
Yes. The key there is usually you can't have completely disjoint sets with regard to the endpoints of interest. So if somewhere in the literature someone has done a trial where they've looked at two of these endpoints in the same trial, then we can use that information to bridge. There's always the possibility of just sort of chucking all the data in there and trying to fit it all with one model, and we absolutely have done that. But it is, I think, helpful to have, for these related endpoints, if possible, a trial where they've been looked at together in the same subjects. Not necessary, but helpful, I guess is the word on that.
How do you know if the literature is rich enough for a model-based meta-analysis? Often it's possible to conduct a simple meta-analysis, but you don't know if there's anything to model.
Yeah, so that's, I mean, I guess, again, I think the first thing we do is, when we are assembling the database, is we start evaluating the feasibility of what sort of models the data might support right off the bat. Even in what we could, a large model-based meta-analysis, oftentimes there may be drugs or classes for which the literature is so thin we can't really estimate dose response, in which case we usually would model that drug as at the treatment level of effect. So it's clearly, this type of analysis is clearly something that you can't be very prospective about. You have to be sort of exploratory in your approach, and then, in evaluating, the sort of breadth and depth of the literature, that's going to somewhat be a guide to what is feasible in terms of the analysis that data would support.
How much computational infrastructure is required to perform this kind of analysis?
Oh, very nice question. Thank you. Unlike population analysis, which works with a lot of individual data, and you're working in a very highly computationally intensive atmosphere, model-based meta-analysis, because you're largely working with summary information, essentially we typically use R for this, and it's usually run from a laptop as opposed to having to run it on a cluster or a grid or something like that. And the models in general often will run in like a few minutes. If there's a really large data set and you're, in some cases we might switch to a mixed effects framework, that can take maybe an hour or so to run, but nothing more extreme than that I've experienced to date.
Is it possible to do model-based meta-analysis with patient-reported outcomes?
I think what this person is attendee might be asking is, can you do individual data in addition to summary data? Oh no, wait. Maybe I think it means that the patient is reporting the outcome.
Yeah,
yes,
I think that's what
yeah, okay, yeah. No, I don't see, there's no real reason that you couldn't do that. In fact, it doesn't really matter, I think, whether it's physician reported or patient reported. I think, again, part of it will depend on how often or how much that endpoint is really reported in the literature, but there's really no need to distinguish from an analysis standpoint between patient-reported or physician-reported. And as a corollary, or at least how I initially interpreted the question, there are instances where it's possible to mix both individual level data with the summary level data in one big analysis. So that's been done in certain instances as well.
Can you comment on differences and similarities between the different endpoints you used in the last example?
The similarities and difference in the last case study example?
Yeah.
Well, I mean, each of the indications used a slightly different measure of efficacy. They're, and being different diseases, they have different scoring and different scales. So this is something you have to account for in the modeling. I'll be honest, I'm not, I don't have an in-depth understanding of how each of those scales is sort of broken down within each of those indications. So I'm probably not prepared to go into detail about that in this particular talk.
When bridging from one indication to others, what are your key considerations besides exposure, dose, and targets?
Yeah, so again, I think one of the things that exposure, dose, and target, I think, as I was saying, baseline status is usually a big explanatory, a big effector of how much response you're going to get. Regional effects may also be important. Oftentimes, it appears that particularly Japan versus a lot of the rest of the world will often have somewhat different results. So I think it's a matter of really exploring your data and kind of understanding what sort of trial design or population characteristics really have an impact on the response. And that, I would probably, if I was looking at that, look at the data within each indication to sort of look for these trends and then see how generalizable they are across the disease states.
How do you typically implement the weighting for sample size in models?
Well, essentially in R, it's pretty easy because basically you can just specify we generally use the GNLS capability, and then within that there is a place you can just specify the weights. Of course, you're going to need a column for, well, either sample size, if you're working with, dichotomous or binary endpoints, or then, as I mentioned, maybe standard deviation in your data set. And then essentially you can read that in and, employ it in the weighting. The same is actually true if you wanted to do this in non-mem. It should be fairly straightforward to incorporate that into your residual variability expression.
Looks like this is our final question from the audience. Is there a benefit to conducting Bayesian-based, model-based meta-analysis with STAN or WinBugs?
I don't see a reason why not to, especially if you wanted to start with a prior and then, if you were getting proprietary data, you could potentially develop a literature model and then use that as a prior and then see how, your results compare to that. In general, for most of the questions that we've had to answer, the Bayesian approach has not really been needed. And so it really, I guess, depends on sort of your particular inclination. But as I said, generally speaking, we've not found the Bayesian approach to be, warranted to answer the questions that our clients have had largely.